Antigens are substances. When they enter into the body, they stimulate immune response. There are two kinds of immune responses. One is antibody production. The other one is activation of T cells. Antigens like bacteria, when they enter into the body, these antigens stimulate the immune system to show the response. Antibody production is a kind of immune response. So in this case, how to define the antigen? A molecule that generates an immune response. Let us see another case. Here there are antibody molecules. Our blood has naturally available antibody molecules. The plasma of A blood group individuals have anti-B antibody molecules. When there is mismatching during blood donation, that means if B blood is given to A blood group individuals, the RBC of B blood group individuals have antigens. These antigens react with the preformed antibodies. That results in antigen antibody reaction. Here, how to define the antigen? Antigen is a molecule that reacts with activated components of immune system, that is preformed antibodies or activated T cells. A molecule reacts with antibodies or activated T cells is also called as antigen. So this is also an antigen. It reacts with already activated components. This is also an antigen. It generates immune response. So the first case is specifically called as immunogen. An immunogen is a substance that is capable of initiating an immune response. Immunogen is nothing but antigen. When an antigen initiates immune response, that antigen is called as immunogen. So all immunogens are antigens, but all antigens may not be immunogens. Haptens are substances that are non-immunogenic. What do the haptens do? The haptens can react with the products of immune response. That is, what are the products of immune response? Antibodies and activated B cells. The haptens react with antibodies and activated T cells. The activated T cells and antibodies are the products of immune response. Haptens are incomplete antigens. When the hapten molecules are combined with a carrier, the hapten molecule will be a complete antigen. Another molecule is there. Many drugs like penicillins are haptens. Let us see about another molecule, that is adjuvant. Adjuvants are added with vaccines to increase the efficiency of vaccine. Vaccines have killed microorganisms or partly killed microorganisms. So killed antigens are inactivated antigens. They cannot show the immune response very effectively. Many vaccines cannot promote the immune response very effectively. So an adjuvant is an ingredient used in some vaccines. What do the adjuvants do? These adjuvants help vaccines work better. When an adjuvant is added to the vaccine, it helps to create a stronger immune response in the people receiving the vaccine. So now we have understood the terms adjuvant, hapten, antigen, immunogen. Antigen is a substance generally that stimulates immune response. There are two types of antigens. Some antigens are exogenous, they come from outside. Some antigens are endogenous, they are self antigens, they are produced inside the body. Exogenous antigens, examples, microorganisms, pollen, drugs, pollutants. Pollen grains act as potential antigens. In some cases they cause allergy. Endogenous antigens are blood group antigens. Antigens stimulate the immune system to produce antibody molecules. Here the central one is antigen. This diagram shows the binding between antigen and antibody molecules. Antibody molecules are Y-shaped structures. An antigen has epitope. Epitope is a part of an antigen. This is one epitope, this is another epitope, this is another epitope. Here various epitopes are found on the surface of the antigen. This is one epitope. The epitopes act as antigenic determinants. Epitope is a part of antigen that acts as antigenic determinant. The antigenic determinants are recognized by antibody molecules. After recognition, the antibody molecules bind to the antigenic determinant or epitope. Epitope is a part of antigen. Then the part of antibody is paratope. Paratope is a part of antibody that binds with epitope. So the paratope has antigen binding site. This is paratope. The paratope has antigen binding site. Paratope is a part of antibody that binds with epitope. Epitope is a part of antigen that binds with paratope. Paratope is called as antigen binding site. Epitope is also called as antigenic determinant. Antibodies are produced in response to the entry of antigen. They are called as immunoglobulins. Antibodies are protein in nature. They are secreted in response to an antigen. Antibodies are produced by a kind of lymphocytes. They are B lymphocytes. During antibody formation, the B lymphocytes become plasma cells. The plasma cells produce antibodies. There are 5 classes of antibodies based on the functioning and the chemical nature. They are IgG, gamma, IgM, mu, IgA, alpha, IgD, delta, IgE, epsilon. During 1950s, based on the experiment, Porter and Edelman revealed the structure of antibody molecules. Antibody molecules are Y-shaped structures. They are made up of 4 polypeptide chains, 2 pairs of polypeptide chains, that is, pair of heavy chain and pair of light chains. Light chains are designated by L. Here this is Y-shaped antibody molecule. It has pair of heavy chain, pair of light chains. The heavy chains, light chains differ in their molecular weight and number of amino acids. Light chain has the molecular weight of 25,000 Dalton. It has 214 amino acids. And the heavy chain H, it has the molecular weight of 50,000 Dalton. It is made up of 450 amino acids. The molecular weight of light chain 25,000 Dalton, heavy chain 50,000 Dalton. Light chains are made up of 214 amino acids. Heavy chains are made up of 450 amino acids. There are 4 polypeptide chains. When proteins have many polypeptide side chains, they are connected by disulfide bonds. Here the Y-shaped antibody molecule has pair of heavy chain, pair of light chain. Here the disulfide bonds. Some of the amino acids are sulfur containing amino acids. They involve in disulfide bonds. This is a light chain, this is heavy chain. Both are connected by disulfide bond. Here also light chain, heavy chains are connected by disulfide bond. And these 2 heavy chains are connected by disulfide bond. So Y-shaped structure is formed. The polypeptide chains are linked together by disulfide SS bonds. Because of the SS bonds, the primary structure of protein becomes quaternary, tertiary. The SS bonds give stability to the quaternary and tertiary and quaternary structure. The protein will be functioning in the particular structure, tertiary or quaternary structure. That functional status is maintained by SS bonds. A bond is formed due to the presence of sulfur containing amino acids. Each heavy chain L chain has 2 terminals. One is C terminal or carboxyl terminal. This is called a constant region. These regions have Similar it is in various antibody molecules. So this is constant region or C terminal or carboxyl terminal. And another end is amino terminal, N terminal or variable region. These regions have variation in different antibody molecules. This region gives specificity to the antibody molecule. A particular antibody recognizes a particular antigen. This is because of the N terminal. The V terminal or variable region. Variable regions are found in both light chain, heavy chain. The variable regions of light chain and heavy chain together form antigen binding site. V regions of heavy chain and light chains combined together form antigen binding site. Antigen binding site is nothing but paratope. Some antibodies are responding to different antigens. Some antibodies respond to different types of different antigens. So they have very different V regions. They have more variations in the V region to recognize different antigens. Here this is an antigen. So antigen binding sites fit a specific antigenic determinant. Antigenic determinant is also called as epitope. Here this antigen has three types of epitopes. One has curved outline, another flat outline, another pointed outline. So the green color antigenic determinant is recognized by a particular kind of antibody molecule. Antigen antibody binding is by binding shows lock and key mechanism. A particular antigenic determinant is recognized by particular type of antibody molecule. Let us see another recognition. The pointed antigenic determinant is recognized by another antibody molecule. So antigen antibody recognition is like lock and key mechanism. In the next part we shall learn about antigen antibody reactions.